So this question is in regards to question 3a, which states, how does the activation state of the receptor affect beta arrestin binding? And what information do the kinase inhibitor studies provide about beta arrestin binding to the receptor? So phosphorylation of the beta-2-AR allows the binding of beta-arrestin to the epinephrine-bound beta-2 adrenergic receptor. When phosphorylation of epinephrine-bound beta-2-AR is blocked by the inhibitor, beta-arrestin can no longer bind to the ligand-bound beta-2-AR. However, when beta-2-AR is not phosphorylated due to the inhibitor acting on the kinase, no band is seen in Western blot when beta-arrestin antibodies are used for immunoprecipitation. This indicates that beta-arrestin will only bind to phosphorylated beta-2-AR, and it shows that phosphorylation of the GPCR is essential for arrestin binding and for desensitization to occur.
Hello, my name is Christy. I'm in Group 6 and I will be presenting question 3b. What is the likely explanation of the differing pattern of protein X phosphorylation in the beta-arrestin depleted cells compared to the control cells? As shown on the figure below in the results of the Western blot compared to the phosphorylated protein X under epinephrine incubation under control siRNA cells and beta-arrestin siRNA cells. On the Western blot, arrestin binding only occurs in the absence of inhibitor, also when the receptor gets phosphorylated. On the control section, we are able to see the decreased levels of phosphorylated protein X due to when arrestin binds the receptor, it triggers the desensitization, in which there is no signal for no cascade will stop. As for beta-arrestin siRNA proteins, then when arrestin is blocked, receptors will not desensitize, in which cascade will continue.
My name is Jose Lopez. I am presenting for question 4, part of Group 6. Question 4 asks to describe signaling pathway of angiotensin II subtype 2 receptor in vasodilation. Part A asks which figures or show figures showing all players of the pathway. We're using our first source, so both the diagram and the information. So first off, we can ignore this little corner that has nothing to do with it, but the rest of the information is pertinent to the question. So AT1R is the receptor responsible for vasoconstriction. AT2R is the receptor responsible for vasodilation. B2R forms with A2R, see B2R right here, A2R, creates a heterodimer. They can are essentially the same thing. They create the same product with the exception of A2R, but we're disregarding that. The GAI or GAI, I guess, is the G protein. ARBs inhibit ATR1 function, so when that's introduced, this will not be working. Nitric oxide, which is NO, is the product of the heterodimer and the singular receptor. It is also a precursor for CGMP and norepinephrine. And CGMP and norepinephrine allow for blood pressure regulation via vasodilation. For the next slide, we are using our second source and our first source. So the part B asks for explanation of activation and inactivation mechanism of all players of this pathway leading to final physiological response, which is vasodilation. So we're using the same diagram. Again, ignore this bottom right quadrant, I guess you could call it. So AT2R is a G protein coupled receptor and acts as an antagonist for AT1R. Both receptors are found in vascular smooth muscles and affect blood pressure through vasoconstriction. That is for AT1R, again, that's for vasoconstriction, and vasodilation, AT2R. When ARBs, this little circle, when ARBs are introduced, AT1Rs are blocked, so they no longer function as intended. Angiotensin II, which is the primary molecule, undergoes a conformational change to interchange with the G protein. The G protein is the GAI with AT2R. Angiotensin II activates many phospholipases to hydrolyze the phospholipid membrane and initiate signaling. So this is part of the second source. The signals allow for the increased production of nitric oxide, which is right here, NO, which in turn lead to increased levels of CGMP and norepinephrine. So these are these two products. Both of those products lead to vasodilation and reduced blood pressure. Part C asks for citations, and again, our first source is this one, and this is our second source. Thank you.